Ok, donc je vais faire une présentation en anglais, comme on avait prévu. Juste avant, donc oui, ce sera une présentation un petit peu sur différents travaux que j'ai faits. Je fais cette présentation parce que je veux postuler au concours blanc de l'INRA cette année avec le CDGP. Donc comme ça vous donne un petit peu un aperçu des différents travaux que j'ai pu faire. Et puis, n'hésitez pas, si vous êtes intéressé par ce type de recherche, à venir me voir. Moi, j'essaye de savoir un petit peu ce qui se fait plus précisément au CVGP pour pouvoir construire un projet qui s'intègre bien. Okay, so I start with the work that I've been doing during my thesis in Toulouse about Hemosporidian parasites. So these parasites are protists, which are often called avian malaria parasites, because they infect mainly birds, and this group includes the agents of human malaria. But it's a very diverse group with tens of thousands of lineages, and they all have something in common, that they have a life cycle with two hosts. One is a vertebrate species, and the other one is a deep-terrain insect. So, because they infect mostly birds, we think that originally the ancestor of this group was a parasite of birds, or even dinosaurs, because it's a very ancient group, probably over 100 million years old. But throughout their evolutionary history, they colonized other hosts like reptiles and some mammals, including humans. So the most famous, the diversity of Hemosporidians is sorted in three different genera. There's other smaller genera. Plasmodium is the most famous one because that's the genus that includes human parasites. And there's also Hemoproteus and Leucocytosum. As you can see, each genus is specific to one family of insect. For example, Plasmodium is transmitted exclusively by Felidae mosquitoes. So during my thesis, I had a lot of blood samples from birds from the Mascarene archipelago, specifically the islands of Réunion and Mauritius, and I screened the blood samples for Hemosporidian parasites. And I found the genera Plasmodium and Leucocytosum, which is a standard result. Hemoproteus is often absent from island systems. I think, if I recall correctly, because it was a long time ago, I think the vectors of Hemoproteus do not colonize island systems. So what's immediately obvious with these results is that the diversity of Leucocytosum is much greater than that of Plasmodium inside the Mascarene archipelago, with 26 lineages for Leucocytosum versus only 10 for Plasmodium. So we wanted to explain this difference in diversity. And so there's three immediate determinants of diversity in an island system like that. You can be more diverse because you have colonized more often. You can be more diverse because you colonized earlier, and so you have had more time to diversify inside the archipelago upon colonization. And then you can diversify faster. So we used phylogenetic analysis to answer these questions. So we built a global phylogeny with all the lineages of Leucocytosum and all the lineages of Plasmodium that we collected in the island, in the Mascarenes. And we put with that all the sequences that were available at the time in GenBank to represent the outside of the system. We estimated the number of colonizations as the number of monophyletic group of island lineages. So according to this estimation, we would have around 11 colonizations for Leucocytosum versus 6 for Plasmodium. So that's the first explanation of why Leucocytosum is more diverse in the archipelago. They colonized more often. I won't get into the detail of the rest, so we time calibrated this phylogeny in relative time. so that we could compare the relative times of colonization of leukocytosome and Plasmodium, and so that we could also estimate diversification rates. And what we observe is that not only leukocytosome have colonized more times, but they colonize on average earlier, so they could accumulate more diversity inside the archipelago after colonization. On the contrary, we could not detect any differences in terms of diversification rates. So now we will remain on Reunion Island. There was a work still in Toulouse for my first postdoc, and it's about a species of gecko, Phexuma borbonica. This species is almost endemic to Reunion Island. There's a small population on the Agarega archipelago, but it's maybe it's a recent introduction. It's considered an endangered species by the IUCN, and the greatest threat to this species is the alteration of its habitat by human activities. As you can see on all these pictures, there's a great variation of color patterns of this species across the island. And so our objective with this study was to look if there is a genetic structure underlying, maybe these populations are differentiated, and try to see if we can identify different evolutionary lineages. And this has notably implications for conservation strategies. So these are the results for the mitochondrial genome. So, I mean, genome is just two genes. This is the mitochondrial phylogeny, and we divided it into, there's two main clades, which we call the blue and yellow clades, which have split from each other roughly around 1 million years ago. Time calibration is not very precise, but it gives you an idea. If you map the distribution of these clades, you see that there is two zones. Basically, you can draw an axis here from the center of the island to the southeast, and the two clades are distributed on either side of the axis. And the localities in red here, you can find both clades. So there is a quite ancient structure that we can observe at the mitochondrial genome. If you look within this clade, there's also a lot of geographic structure. A lot of sites have only private haplotypes, and most of the haplotypes are found only at a single site. So there's quite an obvious structure with the mtDNA. We also use microsatellites to look what happened at the nuclear loci, to have the standard structuring clustering analysis here. What's obvious is that these two populations here, dimethyl and milo, really stand out. They are very little mixed, so they seem very singular genetically. These are two mountain populations, here milo and here dimethyl, that are quite isolated geographically from the other populations, and which live in very different habitats from the other populations. Mapping the 5 identified clusters on Reunion, you also see that geographically it makes sense. Also, we tried to compare the extent of differentiation with other groups. So we looked at the FSTs calculated for similar data in other Phexuma species. And the FST that we have in Phexuma borbonica are really high, especially considering the geographic distances between sites. These FSTs are very high compared with the FSTs of other Phexuma species. I'm not getting into the details, but we did a phylogeographic analysis, and combining the data from all markers and our analysis, we were able to propose a phylogeographic hypothesis. So the evidence points towards an ancestral population here in the north, which actually agrees with geology. because basically the island of Réunion is very active volcanically. So during the last million years, basically all the parts of the islands have been covered by lava at some point, except here in the north, there are areas that have not received lava for a very, very long. So according to this scenario, we would have basically an ancestral population in the north, and then two expansions on either side of this axis here. So we hypothesize that this constitutes the main split, the main differentiation, and then we observe significant traces of gene flow, especially at the south of the island here. So kind of like a secondary contact. Overall, the data suggests that the overall level of gene flow has been low for a long time. We also, using microsatellites and coalescent analysis, we estimated effective population sizes, which are overall pretty low, especially for the three mountain populations here, especially for population Dimitic here. So in conclusion, Flexuma borbonica is composed of different evolutionary lineages, even if we cannot make really discrete categories, that have been evolving for a long time, quite independently, not very completely independently, but with low levels of gene flow. Also, we observe quite small effective sizes, especially for the mountain populations. So this is something that needs to be taken into account for conservation purposes. For example, translocating individuals from one population to the next is probably not a good idea. So then I went to Groningen in the Netherlands to work with Rampal Etienne on phylogenetic analysis.
Vous pourriez couper les micros, s'il vous plaît.
Merci. Work. So that's when I started to really get into the methods. So I went to work with Rampal about a method based on likelihood for co-phylogenetic analysis. So basically, co-phylogenetic analysis is when you have a phylogeny for hosts, a phylogeny for parasites, and you want to estimate their joint evolutionary history. So to reconcile the phylogenies of hosts and parasites, you need to evoke a series of events. For example, you can have duplication of the parasites within a host. You can have, of course, host switches, co-speciation, or you can, some hosts lose their parasites. So at the time, it was, when I did that, it was around 2015. At the time, the state of the art in co-phylogenetic analysis was the reconciliation analysis based on maximum parsimony. So basically, the data are a tangogram, a phylogeny of hosts, a phylogeny of parasites, and the present time associations. And from that, you estimate a reconciliation, which is basically the mapping of the parasite phylogeny onto the host phylogeny. The problem is that oftentimes the softwares would give you as output one reconciliation or a handful of reconciliations without any measure of how confident you can be in these estimated scenarios. And because you don't have also a likelihood function, you cannot do hypothesis testing. So our idea was to develop a likelihood-based approach for this problem. So we designed a likelihood function for reconciliations given a tangogram. To do that, we used rates for the different types of events: duplication rate, host switch rates, rate of losing parasites. And this allows you to calculate the likelihoods for the different reconciliations. But we didn't want, we thought that probably estimating the reconciliation was a very hard problem. Maybe the information contained in the tangram is not so informative about precisely which reconciliation is the best. So what we did is we used the marginal likelihood approach. We didn't try to estimate the reconciliation, and we focused on trying to estimate the rates. So basically, you just use as a likelihood function the sum of all likelihood functions for all possible reconciliations. Of course, there's an infinite number of reconciliations, but you can actually do this sum efficiently by using a pruning algorithm, which is standard in phylogenetics. So just by doing one pass through the phylogeny of hosts, you carry out this huge sum efficiently. So in the end, this approach focuses on estimating the rates rather than the reconciliations. Because it's based on a likelihood function, you can quantify the uncertainty. You can do hypothesis testing if you want to fix some rates to some values or impose constraints to the reconciliations. Also, it can handle multiple hosts per parasite and multiple parasites per host, which was not always the case at the time for the reconciliation analysis. The drawbacks, the limitations of the method, we were not able yet to include post-speciation because it brought up quite a lot of computational problem. And also, even though we can carry out this summation here, it remains hard to compute, and after with more than 10 hosts, it becomes difficult to handle. The time of computation really goes exponentially at a fast rate with the number of hosts. Then I went to Madrid and Tenerife to work with Alexandre Martin and Brent Emerson. And we wanted to infer basically the main migration routes in the Canary Islands. And to do that, we wanted to estimate this migration rate using the structured coalescent. Basically, the structured coalescent is an extension of the standard coalescent model to multiple populations. So you have, each population has its own effective population size, and there are migration of individuals across populations. Brent Emerson had the data set for 200 coleopteran species from 4 islands in the Canaries. And our goal was to estimate for each species the migration rates across the islands. And then by compiling the results for all the 200 species, try to determine which were the main migration routes. But we ran into problems, methodological problems. This is an example. Here you have the phylogeny for a species of Megarthrus. The colors here give you the location of tips on the 4 different islands. So when you see this phylogeny here, obviously there's almost no phylogenetic signal for geography. So when you see this picture, you would think that you cannot estimate the ancestral states with the method. There's just no signal for geography. And you should just infer that migration rates are high compared to the time scale you're considering. But you see here that you estimate with very high confidence that the ancestral location of this species was a hero island. Also, if you look at the estimates of migration rates, all the migration rates from a hero to other islands are very high. So this suggests that a hero is a source. And also the estimated population size for a hero is very low as compared to the other islands. So to us, this did not make sense, first because we don't see how You see, there's only 2 sequences for egg hero here, scattered across the phylogeny. Why should the ancestral location be egg hero? And it didn't make sense biogeographically that egg hero was a source population. So I decided to dig a bit to try to understand what was the problem with the structured coalescent. What I did discover is that the likelihood function for the structured coalescent is multimodal, and you have 1 mode per location. So here in our problem with 4 islands, 4 locations, we have 4 different modes. And basically your analysis, your MCMC chain can get stuck in 1 of the modes. So the result that we saw, we were inside the egg hero mode. And inside the egg hero mode, all the ancestral locations are egg hero. Egg hero has a low population size and is a source of migration. But if you play around with the initial parameters, for example, you can get your chain stuck in another mode, for example, Lagomera mode, which is the same, but for Lagomera. So this is really something that is worrying about the structured coalescent. And I'm in contact with other researchers, Dubé and Maïlis Guélian, who have also found a series of problems with inference with the structured coalescent. And we don't exactly know yet what's happening. What we can say is that some data sets are more prone to yielding problem. For example, if you have a small sample size for 1 location, then it's more likely that you get into the modes of this location. We want to try if maybe the priors that we used are not appropriate because they allow very large differences in migration rates. And I've been scrutinizing the model, mathematically for mathematical problems, but so far I just never found any problem with this model. To me, it seems correctly defined. Now, the rest of the talk will be about what I'm doing right now at ISEM with Josselin Cornuault and Fabio Pardi. And it's about phylogenetics. So phylogenetics is basically phylogenetic analysis when you have collected samples from different points in time. And it's especially used in epidemiology when you have different samples collected in the course of an epidemic. So I will first present the 3 models that are mostly used in phylogenetics, and then I will expose a new method that we are developing and now testing with Josselin. You have 3 main approaches to 3 main models for phylogenetics. So these are 3 models: the birth depth with sampling, and 2 versions of the Kingman coalescent, which I call the skyline and the mechanistic approach. The birth depth sampling BDS model has 3 parameters: curve for the transmission rate through time, a rate for SI, and a rate for sampling. Coupez les micros, s'il vous plaît, merci. So,
Oh putain.
So basically, what's the phylogenetic tree in epidemiology represents a transmission tree. So one branch of a transmission tree is an infected individual, and like Hector in this case. And when Hector transmits the parasite to Marie, then this creates a node in the phylogeny. This creates a node in the phylogeny, and this happens at rate lambda. Also, at some point, Hector may lose the parasite either because he recovered or because he died. And this creates a tip in the transmission tree, and this occurs at rate mu. So with these two kinds of events, the BDS model is a model for the complete transmission tree. So the complete transmission tree includes all the individuals that were ever infected by the parasite in the course of the epidemic. Of course, you cannot access the complete tree, but what you do is you collect genetic samples at different points in time. And this gives you, from these genetic samples, you can have an estimate of the sampled transmission tree. So that's the part of the complete tree that links your samples. And the BDS model requires that this sampling process is modeled. And it assumes that it's modeled at rate, that samples are taken at rate psi of t. The main advantage of the BDS model is that you can, the parameters of the BDS are epidemiological quantities that are already meaningful. Lambda is the transmission rate. That's something interesting. Mu is the rate at which you lose infection. So it gives you the average duration of infections. That's also interesting. And from these parameters, you can also deduce estimates for the prevalence through time and for the reproduction number through time. The drawbacks are that you need to assume a sampling process. So you need to parameterize this function psi of t when you do inference. And we will see right after that a wrong choice of parameterization can have large impacts on the results. Also, since 2021 and this paper by Luca and colleagues, we know that the parameters of the BDS are not identifiable. By this is meant that if you give complete freedom to your free function, lambda, mu, and psi, that they can vary in whichever way, then the information contained in a sample tree is not enough to estimate these different functions. So that means that either you have to make more assumptions about the shape of these functions, or you need to bring in additional data. Here we see an example where the BDS fails. In this case, we have simulated an epidemic. You see here is a population size through time in black. So that's the simulated data. And in red, it's the inference made with the BDS. And you see that you have an accurate estimate of the population size through time. But here we have simulated, we have taken samples at a constant rate. And we also have done inference with psi, a constant function. But now in this example, it's the same population size. But the difference is that we have taken samples at a rate that is not constant at all. And we still did the inference assuming a constant rate. And you see that when your parameterization of the function psi is incorrect, the results are completely off. So this means that there's a high sensitivity of the BDS model to the parameterization of the sampling process. The second most used model in phylogenetics is the Kingman coalescent. It has one parameter, it's the instantaneous effective population size. The coalescent does not make a lot of assumptions about the process that generated the complete tree. It's known to be robust to the tree model because it's basically many population models that generate trees at large population sizes converge to the coalescent. it doesn't make any assumption about the sampling procedure because it conditions on the sampling times. And basically the way it works is quite simple. Any two lineages in the sample tree coalesce at a rate that the inverse of theta. So you need then to assume some parameterization for this function theta. And you have two main ways of doing that, the skyline and mechanistic approaches. With the skyline approach, theta is represented by a phenomenological function, like a piecewise function. Here you have different times intervals, and theta is led to very freely long intervals. The advantage of this is that because you use the Feynman coefficient, you don't make any assumption about the sampling process. And the drawback is that your parameterization of theta does not include any parameter, epidemiological parameter that you may want to estimate. So you cannot estimate lambda, mu, or even prevalence through time. Oftentimes, the function theta is amalgamated with the population size through time, but it's not necessarily true that the two are proportional. We see an example of this right now. So we simulated here the population size in black, and in blue you have the simulated theta. And you see that the curve for theta is not proportional to the curve for the population size. So here you see that the estimate of theta with the skyline is accurate, but it doesn't represent the population size. With the mechanistic approach, you still make this assumption of proportionality between theta and population size, but you assume that population size is as predicted by an epidemiological model of your choice. For example, the SIR model is very popular in epidemiology. You have three kinds of individuals: susceptible, infected, or removed individuals. And oftentimes you assume that the total population size is constant. This SIR model is defined by a set of differential equations. Importantly, these equations include these parameters that are meaningful epidemiologically. Mu, as before, is the rate of losing infections, and beta here is related to the transmission rate. Therefore, by solving this system of equations, you obtain the curve for the number of infected individuals through time, which is actually parameterized in terms of the epidemiological parameters. This allows you to have theta to be dependent on epidemiological parameters. So like before, because it's based on the Feynman coalescent, the mechanistic approach does not assume a sampling process, and you can estimate, in theory, epidemiological parameters. Now, as said before, it assumes that theta is proportional to the census population size, which is not generally true. And it's also sensitive to your choice of epidemiological model. We have seen here an example with the SIR, but you could have chosen another type of model. And here is an example where we have simulated the data with an SIS model. So the SIS model predicts that population size reaches a plateau and stays there in equilibrium. So you see the simulated curve in black goes up and then stays at a plateau and oscillates around the plateau. And we fitted an SIR coalescent model with this data. And you see that the estimated curves really all go up and down. So we have a very strong confidence of this method that the epidemic is presently recessing. Okay. But it is not what is happening. The true answer is that the epidemic is plateauing. So now we outline this new method that we are presently testing with Céline. And we predict that it will be the following set of advantages and drawbacks. As you will see, it's also based on the Kingman coalescence, so you don't need to assume a sampling process. You don't need to specify the exact model that generated the transmission tree. It is parameterized in terms of these epidemiological parameters that we want to estimate. The important drawback is that the sample, like for the BDS model, the sample tree is not enough information to identify the parameters. So we really intend this method not as a standalone phylogenetic method, but to be used in conjunction with auxiliary data that are informative about the parameters of the model. So we will see right now what we're talking about. So we make the following assumptions: that the epidemic unraveled according to birth-death type model. The only thing we mean with that is that you cannot have multiple transmissions at the same time. So basically, we assume that the generated transmission tree is dichotomous. Also, individuals are assumed to be exchangeable. So at one point in time, every individual is the same transmission rate, same recovery rate, and no structure in the model. Then the result that we obtain, so that's here really put in a nutshell. We found that provided population size is large enough, the probability distribution of a sample tree under any model of the birth-date type converges to the probability distribution of trees of the Kingman coalescent with this specific parameter. So theta is the population size divided by twice the transmission rate. Then we parameterize the population size with this function here. So we make the population size dependent on the rates of losing infections and transmission rate. I will not get into the detail, but we have a justification to say that this is a universal expression of population size based on rates of birth and death. So what's important here is that we have relatively few assumptions, but we still can express theta as a function of important epidemiological parameters. Transmission rate, rate of losing infection, and not is the prevalence at the present time. Same problem as for the BDS, like I said before, these three parameters and not lambda mu are not identifiable. That is to say that you can find infinitely many values of these different parameters that give you the same function theta. Hence the auxiliary data. So to sum up what we're trying to do here, you have a sample tree from this. Using the Kingman coalescent, you can estimate theta. We parameterize theta thanks to a mathematical result. We obtain this expression for theta based on the epidemiological parameters that we're interested in. And from these parameters, you can also deduce the curve of population size. You can deduce the reproduction number through time. If you do this analysis alone, you can't estimate your parameters. So you include in the same analysis some of the types of data which we will be seeing examples in a moment. And this calibrates some of the parameters and renders the things identifiable. So as a first preliminary test, what we did was fixing the function mu to the true value that we used for simulations. It's some way of mimicking auxiliary data that are very informative about the death rate. So you recall this example we've seen earlier of the BDS model that fails when it assumed the incorrect parameterization of the sampling rate function. We did the same with the same data set. We used our method, and you see that the estimate of the population size and of the reproduction number are accurate. So sometimes you have a high uncertainty, low precision. It's in general because you have a few coalescent times in these time points, okay? But you see that it's still accurate. We have seen this example of the Sky9 approach, where the population size was not proportional to theta. In our case, we still can estimate the population size accurately. And same for the reproduction number. And the final example that we've seen with the SIR model, we use the same data and fit a new method, and we again obtain accurate estimates of population size, reproduction. So that bodes well for the use of the method, but really what we want to do here, we fixed the rate, the death rate to some known value. We cannot do that in practice, so we need to invoke auxiliary data informative about some parameters. So here are a handful of examples that we are going to be testing as potentially informative auxiliary data. Okay. For example, if you work in humans, you can, by contact tracking, we can determine when an infected individual approximately was infected and when he transmitted the parasites to other people. From this information, you can inform lambda and mu. You can try to measure durations of infections. So you follow an individual and you see when they recover. This gives you information about the rate of recovery mu. You can have data, for example, at time 1, you follow a sample of infected individuals, and at time 2, you see how many have recovered. You can use incidence data, like the number of new infections in a time period, that gives you information about the product of lambda and population size. Or you can do a prevalence testing, so you take a random sample of individuals and you see how many are infected. So that's the kind of data, it's not settled yet, but the kind of data that we are going to consider. And we see we're starting the tests right now, we see. So to sum things up, this is the idea. If you have the sampled phylogeny alone and you use a method without auxiliary data, you basically have very high uncertainty. Okay. And then as you start to include auxiliary data for some time points, you start to reduce this uncertainty. Importantly, we think that if you include auxiliary data at some discrete time points, thanks to the phylogenetic analysis, the uncertainty reduces also in between the time points. Okay? So it comes really as a calibration of the parameters of the phylogeny, and then the phylogenetic analysis takes care of the rest. And we will try to evaluate the robustness of our method to violation of the assumption that we've made, what happens when we include geographic structure in the simulations, or when we consider epidemics with different types of individuals, like in multi-stage infections or with different variants, etc. So thank you very much for your attention, and I will take question.